Welcome to the second lecture on epilepsy. Today, with this lecture, I would like to focus on the process of epileptogenesis. I am Professor Latika Mohan, Professor and Head of Physiology, AIIMS Rishikesh. So let's see, how does a seizure occur? We start with focal onset seizures. So focal seizures basically originate from a hyperexcitable group of neurons, a small group of neurons, maybe about 1,000 or so. We can't really predict what number, but 1,000 seems to be a reasonable number. And these neurons have enhanced excitability. Now this enhanced excitability may be due to certain altered cellular properties like the cell membrane channels, etc. may be more excitable, or they may be arranged in re-entrant circuits. Special synaptic connections may be there amongst these neurons, so they perpetuate their own excitability. And what kind of clinical symptoms this focus is going to produce or lead to depends on where it lies. So the focus, if it's present in the frontal part of the brain, it may create thought disorders to start with. If it is present on the occipital part of the brain, which is near the center for vision, you might get visual hallucinations to start with. If it's in the temporal side, there may be sounds which start with. And then, of course, the various other manifestations of epilepsy which occur. So, how does a hyperexcitable focus lead to the development of a full-fledged seizure? So, there are a sequence of events or a process that occurs which is hypothesized. There is a group of neurons, as I already said, which is the hyperexcitable, and it forms this focus. Now, surrounding this, there is a surround inhibition which prevents from the focus from constantly producing epileptic seizures. So there is always a walling off or a surround inhibition which keeps this excitable area under control. But there is some process which leads to the synchronization and breakdown of the surround inhibition, and that in a kind of way that hyper excitability leaks out of the focal area, and that causes the seizure to spread. It moves down the normal tissue, the normal pathways, and involves the entire brain and leads to a full-fledged seizure. So something like this diagram, this is kind of a schematic diagram. In any area of the brain, there may be a small group of, say, about 1,000 neurons which are hyperexcitable because of their own properties, synaptic connections, or their channel and membrane properties, and they kept walled off by a group of other neurons which are surrounding, which may be secreting an inhibitory neurotransmitter like GABA. So this surround inhibition keeps this focus under control under normal circumstances. But if the milieu is right, if the circumstances are right, sometimes this electrical activity which is abnormal tends to leak out of the surround inhibition and spread to the other tissues, leading to that seizure to happen. So what is this excitability or hyperexcitability that exists amongst these neurons? There's a peculiar kind of biophysics kind of electrical activity that goes on, which is a large, long-lasting depolarization. So a normal action potential is over in a couple of milliseconds, and it is about the same size otherwise. So this would be a large depolarizing kind of drift of the resting membrane potential. And at the peak of this drift of this resting membrane potential, there is a trigger of or series of action potentials which happen. And after this action potentials are over, these hyperexcited, there is a after hypopolarization or increase in negativity or depression after the episode is over. So this diagram would kind of explain what I have just been talking about. See, if you see the bottom of the diagram, you have this is the resting membrane potential, and it is undulating in a kind of a sine curve. At the peak of this depolarization or move towards the positive side, there is a train of action potentials that occur, and this leads to eventually a calcium activated increased potassium conductance, which leads to a hypopolarization, where there is a period where the membrane is not sensitive and is not able to give off any spike potentials. So this particular configuration is what is seen as an intracellular recording. If you see it extracellularly, you would probably just pick up the action potentials. And if you see it in the surface EEG, it comes as a surface spike, which is, so if a spike is seen or a sharp wave is seen in the EEG, it is basically meaning that underneath it there is some kind of activity of hyperexcitable neurons which is going on. Now this may just be isolated in one area, walled off by that surround inhibition, or it may spread and create what is known as a seizure, where this gets into the rest of the tissues, and it is, once it gets into the rest of the tissues and spread, there is a period of kind of a depression which happens later, which leads to the negative symptoms of the seizure. So what basically characterizes this paroxysmal depolarizing shift? The intrinsic membrane properties, the properties of the neurons which are there in that, they have certain types of channels which are present in them, which predispose to this hyperexcitability. Then there may be activation of certain very highly excitable, hyperexcitable receptor channels such as AMD, PA and NDMA, glutamate receptors, and the removal of a magnesium block and synchronization of this group of neurons together. There may be increased synaptic input from various excitatory neurons which may feed into this group of neurons, and there may be development of abnormal synaptic connections, interconnections between these neurons. So they kind of keep feeding into each other and causing more and more re-entry circuits which hype up the excitability further. So long as this kind of abnormally channel properties of those neurons and the interconnections and this excitatory neurotransmitter feeding into it, it remains inside that local area only. There is no clinical manifestation. A person may be having this all the time, but he may not have any kind of abnormal symptoms. But the moment there is something that breaks down the surround inhibition, which may be a change in the excitability of the entire cortex, it may be due to sleep deprivation, it may be due to increase in temperature, it may be due to various other problems in the milieu interior of the body like alkalosis or acidosis or anything like that. When that surround inhibition breaks down, then this increased excitability leaks through. It leaks through. So this surround inhibition, it due to either the intense firing during depolarization shift or failure of the GABAergic inhibitory controls. When the surround inhibition is overcome, the seizure begins to spread, and when this happens, it is unpredictable. So as I said, it is dependent upon hormones, menstrual cycles, sleep shifts in glutamatergic activity, GABA activity, the milieu interior, and so many other factors. So when the surround inhibition starts to break down and the electrical activity, that is hyperactive active action potentials which are coming out of that focal area, when they spread, the first to be affected are the surrounding normal tissue. So there is this abnormal tissue in the middle, and then there's this surrounding tissue which is normal. So when the surrounding tissue gets excited, wherever that focus is, those kind of symptoms start to happen. So if it is in the temporal lobe, where there is the area for hearing, a person may start hearing some abnormal weird sounds, and that may be heralding the beginning of the seizure. Similarly, if it is in the visual cortex, the focus is in the visual cortex, the aura may be in the form of seeing various lights and hallucinations. And generally the seizure, if it is slow, if this electrical activity abnormal is spreading in a slow fashion, there may be a series of stereotype clinical manifestations, just as the hand may be moving in a particular stereotype way, or there may be smacking of the facial muscles, the mouth may be opening and closing, or blinking of the eyes, depending on which area is getting excited step by step. And that is what we call the Jacksonian march. and it's a typical feature of focal epilepsy. If the whole cortex gets overwhelmed, especially the motor areas, the entire musculature of the body goes into a tonic spasm. That is known as the tonic phase of a generalized seizure. So, the acute muscular contraction, including the laryngeal muscles, also contract, which gives rise to a cry. They were also that causes what is known as the tonic phase of a epileptic fit or a seizure. And immediately after the tonic phase, everything relaxes, and then it may go into a kind of a multiple spasm and relaxation phase, which is known as the clonic phase of the seizure, and hence we call it the tonic-clonic phase of the epileptic seizure. Okay. So termination of a seizure, there may be after an intense phase of excitation like this, which may last for about 30 seconds, complete exhaustion of all neurotransmitters takes place, and there may be there's a metabolic exhaustion of the ATP. There is everything gets exhausted in the brain, and that leads to a kind of a phase or negative phase of loss of consciousness and a period of disorientation or confusion, and there may be even paralysis of one part of the body or some other blindness or something which resolves over a period of time, and this is known as this post-ictal meaning seizure, post-ictal confusion. So generalized onset seizures, this is slightly different from what we have just discussed about focal seizures. In generalized onset seizures, there is a sudden catastrophic onset. So you don't see any aura and there is no build-up. Generally, the cause of generalized seizures is a group of abnormal channels which may be present towards the central portion of the brain. So this may be located in the thalamus or some other subthalamic, subcortical nucleus. And this group of channels basically gives the initial sort of trigger to the excitation, and so it may involve both hemispheres from its very onset, and it may not have any it's just a on-off kind of thing. There may not be any post-ictal confusion. So there are two main hypotheses. One is that whole cortex itself is hyperexcitable. So the seizure happens because of the cortical hyperexcitability for various regions, which may be related to neurotransmitters, channel properties, may be related to the milieu of the cerebrospinal fluid and the extracellular fluid in the brain. And the thalamic origin, that means there are certain triggering channels which are present in the thalamus, which gives the initial trigger to the already hyperexcitable cortex and leads to this catastrophic bilateral sudden kind of a epilepsy seizure that happens. So both the hemispheres are disrupted simultaneously. There is rapid synchronous activation of all the normal pathways, and that is what leads to a generalized seizure. So let's try and figure out what is this epileptogenesis. Now, the process by which a normal brain is functionally altered and biased towards the generation of abnormal electrical activity, and that is what forms the basis of any chronic seizures or epilepsy. So the traditional anti-epileptic drugs that we've got, they can terminate a seizure, or they can control future seizures and keep them keep a person from having more seizures, but you cannot prevent the process of epileptogenesis. That means when you give an anti-epileptic medicine, you're not curing the patient, you're basically just keeping the epilepsy under control, and the moment the drug is stopped, again the problem will start. So what do you understand by epileptogenesis? It is in three phases. So there is an initial precipitating event or injury which may damage the brain. It may be in the form of trauma, it may be a formation of a clot like in a stroke, it may be the development of a tumor, or it may be an infection like cysticercosis. So these are basically the primary event which would lead to an abnormal focus from developing. Then there is a latent period during which the injury transforms this normal tissue into an abnormal focus. And this is the period of interest of all current research, where you can intervene and prevent that injured part of the brain from becoming a focus of abnormal electrical activity, which would give rise to recurrent seizures. So this is where you can do something and prevent the state of epilepsy from developing at all. So what are the various processes in epileptogenesis? There is inflammation, there may be some cell death, loss of certain inhibitory neurons, and activation of certain molecular signaling processes, which I'll cover later. Certain new genes may be expressed, and maybe expression of certain new ion channels which are more hyperexcitable, and there may be the form this axonal sprouting, that means new connections being developed amongst the neurons. So especially these new circuits which are self-sustaining and excitable, which are formed pathological, excitatory, recurrent, abnormal synaptic connections, which lead to developing that hyperexcitable group of neurons which are more excitable and they're able to sustain the excitability. And that is what is the formation of an epileptogenic focus. The pathways which are under study right now regarding this are the BNDF, the TRKB signaling pathways, the mTOR pathway, and the REST pathway. These are under active research as possible ways by which this epileptogenesis happens. Now, certain epilepsies with genetic etiology, there are about 40% of these which have a genetic background. They may be due to a family inheritance or due to genetic mutation. So either they inherited or it may be due to a specific mutation in that particular person. They may be for genes which encode for certain ion channels. They may be genes which are assisting abnormal neural migration. So that means maybe some area where the gyrus may not develop, or there may be a reduced development of certain gyri. Various cortical malformations which may act as epileptogenic foci. So basically abnormal connections of the neurons or abnormal channels which are present in some of the cells. That's the bottom line as far as these are concerned. And there may be certain excessive excitatory circuits which are feeding onto them, or there may be loss of inhibitory circuits. And there's another variety where certain genes may cause biochemical changes which overall influence cortical hyperexcitability. So these are the ones which are leading to the genetic type of epilepsy. Another interesting concept which is coming up in epileptogenesis is that there may be actually a convergence of acquired and genetic mechanisms in epilepsy. So a person who has got a tendency to have epilepsy because of certain genetic problems, if he's put in the right kind of environment, say because of temperature variations or repeated febrile seizures, the person will be able to express certain kind of proteins, or may be able to initiate a certain secondary cascade of events which leads to establishment of epilepsy. So there is a possibility of convergence of genetic and acquired mechanisms which may lead to epilepsy, which need also further focus on. So there are a lot of questions, more questions than there are answers in epilepsy. There are very large number of anti-epileptic medicines which control the seizures, which prevent them from happening, but they do not remove the cause. Moment you stop the anti-epileptic medicines, again the person will start having the seizures. But we have to look at how we can have anti-epileptogenesis, and we can prevent the establishment of a seizure state in the person at all. And future research directions are towards more genetic manipulations, epileptogenic signaling pathways which I talked about, and potential molecular and cellular targets for various drugs to prevent conversion to chronic seizures and development of epilepsy. Some of the references which I used to prepare this talk and some interesting further reading, please do go through it to understand the topic in greater detail. Thank you.